The overall goal of the following experiment is to test the functional status of mitochondria in living cells and isolated mitochondria by measuring multiple parameters of oxidative metabolism using the XF96 extracellular flux analyzer. These parameters are basal respiration, ATP turnover, proton leak, and maximal respiration or spare respiratory capacity. This is accomplished by sequentially adding specific modulators of mitochondrial respiration to C2C12 Myoblast cells and measuring the change in oxygen consumption rate, or OCAR, an indicator of respiration. The conversion of glucose to lactate, which generates ATP through glycolysis, the other major energy pathway in cells, is also measured as ECAR, or extracellular acidification rate. Mitochondrial profiling results can be obtained that provide insight into the central role that mitochondrial function plays in determining the lifespan of cells.
Hello, my name is David Nicholls. I've been a mitochondriologist all my career, and the most important piece of equipment I've used in my studies has been the oxygen electrode. If you can measure the rate of oxygen uptake under different conditions with isolated mitochondria, you obtain a mass of important information. When you move to cells, especially cells which are cultured in a multi-well plate or on a cover slip, classical methods don't work. This is a major problem which was overcome a few years ago by the development of a multi-well plate assay, enabling us to study the respiration of cells in parallel in a multi-well plate format. What you're going to see today is a very simple but information-rich experiment which we devised in order to monitor basic mitochondrial bioenergetic function